So we're gonna get started. So, like I said, first up, we have the Georgia Aquarium. Our first guest is Ai Ajavan. He is an environmental educator at the Georgia Aquarium. He's engaged in the Society for Fresh Sciences, which is SFS, Emerge program, and serve on the SFS Emerge Scholarship Team for Research Engagement, Advancing Minority Voices, which is also known as STREAM. Before his undergraduate studies at Georgia State University, IE worked on reintroducing endangered fish species with the Tennessee Aquarium and conducted research on fish ecology and anatomy. He is joining us to discuss the role of technology in conservation research. So please give a warm welcome for IE. It's all yours, IE. Welcome.
Thank you, Babita, for that introduction. I love your energy. I love how excited you are. I hope everyone tuning in today is as excited as you are. STEM is a great topic. It's utilized all across the world in all kinds of different areas. But today I'm here to talk about STEM and STEAM at Georgia Aquarium, some of the work that we do with our partners. So to get started, my name is Ai. I work in the education department here at the aquarium. Most of my job is working with students hands-on. We take them around the building, show them all kinds of different parts of the building, but also connect that with what they're learning in class, in their schools. But today I'm here to focus on technology that is utilized in research and conservation. So if you haven't been to the aquarium before, we were the largest aquarium in the world when we opened in 2005. We've now been demoted a couple spots. But we are now the largest aquarium in the Western Hemisphere of this world. With over 10 million gallons in our galleries, we hold more than 600 species of animals. And it takes a humongous team to be able to run this building every single day. We have 600 staff members and over 1,200 volunteers. And our volunteers, by the way, have put in over 1 million hours of their time into this building to make sure that everything is running as smoothly as possible. But everyone is spread out across 24 different departments. And of those, almost 2,000 people, only about 100 of us actually work hands-on with animals every day. I get that opportunity maybe once or twice a week, but we are very, very thankful for everybody in this building who helps, keep everything running as it should be. We have 6 major galleries. First is our Dolphin Coast. This is where our dolphins are, our dolphin presentation. That's where our dolphin trainers work as well. And we have River Scout. This is more of our freshwater gallery. It has many, many different exhibits and tanks that display freshwater fish from around the world, ranging from here in Georgia. We have a native stream tank, so showing off some of the native fish from our area, but also we expand all around the world. We have fish from Asia, from Africa, and even the Amazon as well. And in our sharks gallery, this is our newest gallery. Actually, it opened just a few years ago, but we've got several species of sharks, as well as this huge school of false pilchards. They're kind of like a very, very small fish, but they form this massive school, and they just swim around in a ball all day long, and just looks amazing. That is probably one of my favorite galleries in this building. We also have Cold Water Quest, which focuses on cold water habitats from around the world, but main focus is off the Pacific Northwest coast. So think like Washington, Northern California, in that area. We have our beluga whales in there, as well as African penguins and even puffins and sea otters. There's all kinds of stuff in that gallery. We also have Tropical Diver, which simulates a tropical reef. This one is centered on the Indo-Pacific. So there's tons of colors in there from our fish, from our coral. I love that habitat too, because we have what we call our dump buckets. So they crash down onto the gallery. They pour out thousands of gallons of water every few minutes, and it just creates this wave effect that looks fantastic. That is probably my second favorite gallery. But the most well-known is Ocean Voyager. This actually was built by Home Depot. They donated lots of money to us to be able to house the animals that our founders wanted here in the building. And that includes our whale sharks and our manta rays. that gallery itself is a huge feat of engineering. It holds 6.4 million gallons of water, not just in the gallery space that you can see, but also in the filters and the pumps and everything that goes into it. This gallery itself was the first to house whale sharks outside of Asia. We currently have two. Their names are Yushan and Taroko. They're 27 and 24 feet long. So they're these massive animals, but it takes so much just to run that gallery. Underneath, we have this, what we call our pump room, and there are tons and tons of pumps and filtration that help make sure that that gallery itself is running well. That pretty much takes up the whole underside of the aquarium. And so now I'm going to kind of transition into the work that we do outside of this building. So recently we have been certified as a Center for Species Survival by the International Union for the Conservation of Nature. That's the IUCN. So maybe some of you have heard of the IUCN Red List. This is the group that designates species as endangered, extinct, threatened, all those different levels. So we've worked very, very closely with them, as well as other institutions, to make sure that we are actually putting the work in that it takes to help conserve the nature around us. But to be a Center for Species Survival, this means that you need to be a regional hub for conservation. That means you have boots on the ground, putting in the work, working with animals, working with governments and non-government partners to make sure that policies are in place, protections are in place for all of the animals that we cherish so much. And we have folks here at the aquarium that do those red list species assessments. So they actually go out to the field, take population numbers, and see if these populations indeed are threatened. And so the way that conservation really works is this triangle of plan, act, and assess. So if we start at the plan section, we need to make sure that we're going out into nature, making sure that we're taking proper count of all of the animals and different threats that are there, and forming a plan to hit specific targets. So if we want to increase a population number, we have to set that target. If we want to increase the quality of an animal's habitat, we have to set specific parameters to make sure that we are, that we have targets to hit. And then we act on those. So that, again, is those people that actually go out to the field and help the habitat. Maybe that is finding sources of pollution and educating the community on how they can stop that. Maybe it is doing some captive breeding here at the aquarium to release animals back into the nature. And then after we hit that act section, we move into assess. So we kind of regroup, see what targets we hit, see where we might have missed the mark, and see what we might be able to do better next time. We go back to that planning section to hit the next target and the next one and the next one. And we just keep going around this triangle until we make sure that all of our targets are hit and that all of these animals are where we want them to be. And so, like I said, Georgia Aquarium was the first aquarium outside of Asia to have whale sharks. This means that we pretty much had to become the experts on whale sharks on this side of the world. So that took lots and lots of time, lots and lots of practice, but also lots of thinking. So people that take care of these animals every day, they start to learn the behaviors. We have vet staff here too that takes really, really good observation of all of our animals. But those people work together. So in our gallery, we have kind of formulated this way to take blood draws from our whale sharks without interrupting their swimming or stressing them out. So divers go in the water, they collect blood samples from our whale sharks and pass it off to our vet team. They run those samples for analysis to make sure that our whale sharks are happy. But out in nature, we want to make sure that whale sharks are taken care of, right? We have them here at the aquarium to show people how magnificent these animals are and connect them to a whale shark, right? But that doesn't really do much if we're not actually doing that work outside the aquarium as well. So off the coast of Taiwan, lots of whale sharks travel around the coast. And Taiwanese fishermen actually have these very, very large nets that are set out in place. These are called set nets, and they're humongous. They're about the size of a small house. And the way that works is that an animal will swim towards the net that's kind of hanging out to the side. They'll hit that net and hopefully turn to the right and go inside of the larger net. And then within here, there are different compartments that kind of allow the animals to separate themselves out. So the smaller fish continue through into smaller and smaller compartments, while the larger ones stay in those larger compartments. However, whale sharks are one of those fish that are endangered, and they're also one of those fish that gets caught in these nets. So we've worked very, very closely with our partners in Taiwan, with the fishermen, with universities, and with the government to ensure that whale sharks have protections set in place. So those protections look like having a set time limit for them to free those whale sharks from the nets. But we also work with all of these partners to use acoustic tagging. So what happens is we put acoustic receivers on these nets. And like I said, the Taiwanese fishermen are already using these nets. So it's not like we're going in there and changing all of their fishing methods and, getting rid of their traditions. We're just partnering with them. So we put these acoustic receivers on these nets that are all along the coast of Taiwan, all around the island. I think there's somewhere about 50 different nets around the island. We put those receivers in place. And then when whale sharks do get captured in those nets, we have already trained some of the Taiwanese university staff to go out and be able to tag those whale sharks before they're released back into the open waters. And this allows us to collect movement data on our whale sharks. So we know exactly where they're moving to, what kinds of areas they go to. And then one of our other partners who also works in the Indo-Pacific is through the Reshark program. So specifically within the Reshark program, there is a project called Project STAR. And this is the goal of restoring zebra shark populations in Indonesia, specifically in Raja Ampat. And this species is critically threatened. So we want to make sure that we're boosting these population numbers, specifically the Eastern Indonesian Oceanus subpopulation is the one that is being hit the hardest by habitat destruction and overharvesting. So what we do is we work with different aquariums around the country. This all started by doing genetic testing. So we tested our individuals that we have here at the aquarium, and so did many others. And we found that we actually do have zebra sharks from that eastern subpopulation. And so these individuals have been selected as brood stock. So they lay eggs on their own. Those eggs are already fertilized. And what we do is just go into their habitat and collect those eggs. And then we raise them, make sure that the larva is developing well enough, making sure it's consuming the egg sac that it uses for nutrition. And then we ship these off to Seattle Aquarium, who is pretty much the host of the Reshark program. They have those eggs there for a while, make sure that they are still developing further. Occasionally, they do hatch at the Seattle Aquarium, but we ship either the juveniles or those eggs out to Raja Ampat for reintroduction. And before they are released, we do acoustic tagging on them as well. I'll explain some of that real quick. But it's very important that we discuss how all of these partnerships are happening, right? So Georgia Aquarium, as well as Zoo Atlanta, who will be joining on this program later, we're part of the AZA, so the Association of Zoos and Aquariums. And so that is this network of zoos and aquariums in America, but there's also a global division of that. So we have these partnerships to make sure that we're able to help each other out in these programs, but also if one specific institution is creating a new gallery, or if they want some fish or some animal that another park has, we're able to go in and kind of make a little swap with that. But for the zebra sharks specifically, how do we track success? So like I said, we do those acoustic tagging. So there are these little tags. It depends on what size of an animal we're tagging. You can have some that are the size of like a AAA battery. There's some that are probably about 5 or 6 inches long. But these can be surgically inserted into animals, specifically fish. It doesn't harm them in the slightest. It doesn't affect their swimming, their feeding behaviors, or anything like that. Or they can simply be attached to their fins. But these work, basically, we go out and we set out those microphones, those acoustic receivers, in their habitat. Like for the whale sharks, it's on the net, so we can track their movement along their nets. But this can also be out in the open ocean or in gulfs and bays, wherever it might be. So we set those receivers out in a specific grid pattern, so we know exactly where each one of them is. And then we insert that tag into a fish. And every once in a while, that tag will emit a specific frequency, a specific sound. And each tag has its own unique sound. And that's so that those sounds or those differences in the sound can identify one specific fish. So no two signals sound the same, because we want to be able to distinguish individual from individual. And so when those sounds are emitted into the water, they hit those receivers, and we can go collect that data later on and see exactly when those sounds were pinged on each receiver, and see when that fish was within range of those receivers. And depending on the scenario and where we're doing that tagging, those receivers have a range of up to 1,000 meters, but it could be smaller, around 200 to 300, depending on weather conditions and things like that. But we also track other types of fish as well. So sharks, most people know about shark tagging. So on the left, I have a picture of a smart position or temperature transmitting tag, also known as spot tags. So these are attached to a shark most of the time. They can be attached, but they send that data, whether it be pressure, temperature, or just position, up to a satellite immediately. But that fish does need to breach the surface in order for that signal to reach all the way up into space and hit a satellite. Unlike the acoustic receivers, those can just be in the water. It doesn't really matter when we go and collect that data. But with the spot tags, they do have to breach the surface, and then it is an immediate transmission up to a satellite. And then on the right, I have a PAT tag, also known as a tow behind mini pop-up archival transmitting tag, which is very much a mouthful. But this just means that tag is attached to the dorsal fin of a shark. It's dragged behind it. It doesn't affect its swimming again. And then after a set time, that tag is made to release from the fin, and then it will float up to the surface, stay buoyant there, and then at some point it will transmit all of that data up to a satellite. And then that tag can go and be collected from the ocean. So switching gears a little bit, stepping away from that tagging, I want to talk about what coral actually is, because most people think it may be a plant, but it's actually a colonial animal. So each individual is what we call a polyp, but they're pretty much able to clone themselves, and they share, they're all able to build a skeleton together, which they all share, and which is what unites them all. And within their skin tissue, their skin is clear, but within that skin, they have what we call zooxanthellae. And this is pretty much just algae. So coral are able to use photosynthesis to get their energy, but they also have those really long tentacles with stinging cells on them, which we call nematocysts. And they kind of just sway out in the water, and they're able to collect plankton and small animals that are floating by. They collect those that plankton and then just kind of fold those tentacles in towards their mouth, and they're able to eat like that. But coral bleaching is happening at unprecedented rates around the world. And this is happening because of ocean acidification. So all of the trains and planes and cargo ships and factories are emitting greenhouse gases. This is causing our planet to warm, but those gases are also slightly acidic. So because when they're in the atmosphere, they're able to diffuse into ocean water, that means that they're bringing that acidity with them. So that makes the ocean slightly more acidic than what any animal has ever been used to. But pollution is also another source. So that could be oil spills, that could be fertilizer runoff, whatever it may be, is also a cause for coral bleaching. But what we're seeing recently, especially this past summer, is extremely high water temperatures. Off the coast of Florida this summer, waters hit about 90 to 100 degrees, depending on where you were. We call this a marine heat wave. And so that heat wave lasted 2 or 3 weeks, and that stresses our coral out. So in all of these situations, when that coral gets stressed out, it's really the zooxanthellae, that algae that lives within it, that's getting stressed out. It's not able to feel comfortable. It's not able to metabolize as it should. So it just kind of says, peace out, and ditches from that coral. And without that algae living in its tissue, that coral is no longer able to get as much energy as it needs. It's not able to kind of use that photosynthesis anymore. So it only has to rely on using those tentacles to capture plankton that's floating by. But just because a coral bleaches does not mean that it dies. So that zooxanthellae can come back and host in that coral again, but it just takes time. But if those stressors are still there, if that heat wave lasts, 2 or 3 weeks, that algae is not going to want to come back. So it takes a while. That stress needs to be gone as soon as possible. But eventually, if it can't handle that stress, if that zooxanthellae does not return, that coral will die. And it seems crazy that something like that would wipe out such a huge community, but coral reefs hold more than, I believe it's 50% of all ocean life. So it is, I'm sorry, it's 25%. So on coral reefs, we have huge diversity in our fish, our invertebrates, so things like snails and crabs. So they act as these hotspots for ocean life, but they only cover about 1% of the ocean floor. But they are very, very sensitive. We're creating changes so fast that these corals are not able to really adapt to those changes. But what we can do is find corals that are more stable, corals that are a little bit more resistant, even if it's just by a hair. We're able to go in, collect some of those corals, and do what we call fragging. So this is cutting them up into small fragments. We dip them in iodine to prevent infection to that coral. And then we epoxy those small fragments onto coral plugs and tiles, and then wait for them to recover and grow a little bit. And during this stage, depending on how small you cut them, they actually do grow a lot faster after being cut. So a piece that is about the size of a dime, if you connect that, or if you set that near pieces from that same coral, they will grow incredibly fast and be able to reconnect and form a larger piece a lot faster than they would be able to just grow to that large size on their own. And so after we frag them out, we work with our partners to go out and create coral gardens and nurseries. So we plant them out onto racks like this, or for different species, we might use what we call coral trees. So these are just metal posts with PVC piping coming out of them, but we're able to attach corals to these and make sure that they have ample water flow and sunlight. And we can keep a close eye on the corals that live on these racks. So divers go out, they make sure that that rack stays clean, making sure that no algae is starting to overgrow the corals. And if there is any algae there, they just kind of clean it off, scrape it away, and make sure that it's as good habitat as possible. But technology is inescapable in our world today. So in coral gardens, there are 3D printed coral homes. It's kind of like that tile idea, but this kind of gives them an already set form to grow over. So it allows them to not have to build that skeleton as much for support. They can focus on just spreading out instead, since they already are supported by those 3D printed coral homes. And here at Georgia Aquarium, we have worked with the Coral Restoration Foundation. This past summer, we built what we call the Coral Bus. So our life support systems team worked very closely with our fish and invertebrates department to build the Coral Bus. And essentially what this is just a trailer with a large tank in it. But you can see there is lots and lots of pumping and plumbing. And that's actually what we use to take corals from human care, or either here at the aquarium or some of our partner sites. We can transport them in these large tanks down to the Florida coast, and so that we can do that replanting a lot easier. It's a lot cheaper too, also for, instead of shipping them by UPS or FedEx, whatever it may be, by, carrier, we're able to just put them in there, make sure that they are temperature controlled, that they do have all of that water flow that they need, and that they get sunlight on the drive down to Florida, and that they are as happy as possible during that transition. And then this isn't one of our partners that we work with. This is just some incredible research that I think is completely fascinating. The Sandin Lab at UC San Diego is able to do 3D modeling of coral reefs. So what they do is they swim in a grid pattern, left and right, and then they circle back and go up and down along a coral reef while they take pictures and videos. And they have software that is able to identify coral species and coral groups. So then they can go back to that video. That software identifies similar groups, similar species, and actually paints them a different color in that video. And then what you're able to do is recreate that. So if you import that data into the software, it creates a 3D model. It's able to identify them. But this allows you to go back to the video and tag specific colonies, specific areas, and allows for better monitoring. So you can go back through that video, take a look at the size dimensions, the diversity of the corals in a specific area. And then you could do that all over again in a few months and see actually how fast that coral is growing. If it is bleaching, what colonies are different, if they're bigger, if they might have broken up by fish populations, whatever it might be. You're able to do a lot better monitoring with this technology than you would be able to just go out swimming and take a picture. This allows you to do those calculations, the size mapping and everything like that. And I think that is an extremely useful utilization of technology, especially in coral conservation. But this also is being used in lots of different habitats as well, not just coral reefs. This could be used in seagrass areas, seagrass meadows, kelp forests. You're able to go in and take these pictures, take these videos, stitch them together and create this model of an ecosystem. And this is something that is groundbreaking. This would not be possible, 20, 30 years ago. So as we advance further and further into climate issues, technology plays a big role in how we're monitoring and starting to fix those issues. But technology is also used in ocean exploration. So as we're pushing the boundaries of what we know and what we can learn, scientists are also using remote operated vehicles, ROVs, and they can explore depths that humans cannot reach. So as you go deeper in the ocean, there's lots more pressure, the temperature changes a lot, and it's really just hard for someone to either go dive in there or for a submarine to be built to withstand that pressure. But these ROVs are completely unmanned. Humans don't have to go into them. In most cases, they cannot. There's no space for them to sit. And all of that space is used for instruments. So cameras, infrared cameras. You can have arms on there to manipulate things in the habitat. There's lights. There might be sonar. There's also definitely going to be water collection. So you can take water samples from different habitats and take them back to a research lab and do analysis on what that water chemistry looked like, what animals might be in that water, at a microscopic scale. So there's lots of things you can do with ROVs. And they are not very invasive either. Many of the images and videos that we have of the deep sea actually come from ROVs. So they allow us to see lots of things that we would never be able to see on our own. And I mean, you can just see that they aren't necessarily humongous. They don't really need to be, but there's a lot that they can do. And we've actually been able to discover deep sea reefs. So corals that don't necessarily need photosynthesis, They're able to just live in the deep sea and use a lot of that filter feeding to be able to sustain themselves. But I really just want to highlight what science is. Science is a very, very highly collaborative field. If there's a scientist that says they did something on their own, I can promise you that there was somebody that helped them with something along that process. Many disciplines of science and the arts are starting to work together to find solutions to our problems. And these disciplines don't have to be super specific. They can be broad too. You don't have to be a specialist to help another type of scientist or another person. And STEAM really allows all of us to participate and feel welcome, regardless of what experience we do have. We're able to bring our background knowledge, bring our experiences to the forefront and work together to find solutions to real world problems that we're all facing. And we can utilize various expertise to look at these problems from many different sides, like I said. And so what comes next? What do I ask of all of you tuning in today? I really just ask that you start to consider and think how you can integrate sustainability into your interests and hobbies, whatever they may be. There are small changes that we all can make to help the planet as a whole, whether that be eating out less, using less styrofoam and plastic bags, or even just reusing those. If you're able to walk somewhere instead of drive, that creates less emissions, slows down climate change just by a little bit. But if we do all of that together, we really do have a shot at helping the planet and helping to reverse some of these issues. And secondly, I just want you to consider how can you use STEAM to help in conservation, whether that's conservation on a large scale, like at Georgia Aquarium, or even if it's just in your backyard. Think about the habitats that you share your land with, right? What animals do you see on your day-to-day? What problems are they facing? Are they using trash to build their nests? Things like that. So just think about how you might be able to help an animal just by making a small change and help protect the habitats that you share with other animals. And with that, I will take any questions. And again, I just want to thank you all for tuning in today and for this presentation. It has been a lot of fun. Babita's energy that she has brought to this project has been absolutely amazing. And so just thank you all for tuning in.